Now, chemical carcinogenesis is one of the biggest chapter in the Casret and Dowell, and one of the most confusing chapter in the Casret and Dowell textbook, because of its implication in the science, as well as one of the area where the maximum research works are being done. That is why the cancer is very important, and it is very difficult to understand also. Now, those who have read the topic on chemical carcinogenesis may understand that it is very difficult to comprehend from the Casret and Dowell textbook unless you are having a background in the carcinogenesis, how the various processes that are involved in the carcinogenesis. So that's why I am starting the presentation with the mechanisms by which or how the cancerous cells are being produced, and to introduce you certain terminologies, because when you read the cassette and all, they assume that you know the background of the topic. They do not explain much about what is an adduct, what is a chemical, what is a mutation, or what are the different mechanisms of repair of mutations. So all these things they are not providing in the textbook. So that's why I'll just brush up with the initial portions, what is actually meant by various terms that we are going to use, or the terms that are employed in the textbook. Okay, first one, cancer is a disease that is characterized. So these things are from the Cancer and our textbook itself. The cancer is a disease that is characterized by genomic mutations, modified gene expression, cell proliferation, aberrant cell growth, and neoplasia. Neoplasia or the new growth. So the term neoplasia means it is a new growth or an autonomous growth of the tissue, which can be benign or malignant. All of you know what is the difference between the benign and malignant tumor, and that is induced by the chemical carcinogenic agents. Okay, and cancer actually describes a subset of neoplasia that represent malignant neoplasms. Many textbook has difference of opinion in the definition of cancer, but still, since we are following the castrate and Dowell, I am providing what are the information that is given in the castrate and Dowell itself. Because if you take the cancer definition, some other textbook, it may not be described as the subset of neoplasia. Then coming to the tumor, again, rather a colloquial term, a lesion that is characterized by swelling or increase in the size may or may not be neoplastic. When we study about the classical science of inflammation, there also the tumor comes. Of course, it is not tumor, just indicate it is an increase in the size. So basically, tumor can be a cancerous or a neoplastic condition or a non-neoplastic condition. Only just describes a visible lesion. Okay, so this type of things are important when the objective type questions are being asked in the DABT examination, how to differentiate all those things, or they may give many options and say whether A is correct, B is wrong, like that. When the options are coming like that, this definition, because it is fully based on the Casret and Dowell, I am giving from the Casret and Dowell. Now the cancer genetics. The cancer genetics. Actually, the cancer arises due to a genetic mutation that is disrupting the cell regulation. So it actually means a cell becomes dysfunctional in cancer. Okay, can be a dysfunctional cell in the conditions like the cancer. Then one important concept that I want to introduce is the existence of many of the people who are attending. They will be knowing the regarding the pro-carcinogenic genes as well as the anti-cancer genes. There are in the body we have various genes that will contribute. When they are activated, they may contribute to the development of cancer, and there are certain genes that are present in our DNA itself, which is actually the protecting genes or the suppressant genes, which uh, help the cell to go back to the normal conditions even when some of the pro-carcinogenic genes are activated. Okay, so that involves an inherited or acquired mutations. Okay, in the So actually, the cancer involves the activation of oncogenes and loss of tumor suppressor genes. So either of this condition or when they are coming together, they can contribute to the development of the cancer. And there can be multi-step accumulation of mutations over time. So a single mutation may not produce a cancer. Multiple mutations that is happening at the multiple sites can lead to the mutations. And there are some cancers which are heritable, especially that is important when one of the allele is affected and the other one, say maybe the genes that is derived from the father may be affected, but that is coming from the mother may not be affected. But when a mutation happens to the genome of the mother, then that may precipitate cancer. That is why some people are more prone for cancer. Even a single mutation can produce cancer in such persons. And the genetic instability of the genome can increase the mutation rate. So the genetic instability, again, one of the parameter that is produced by the xenobiotic agents. Okay, we'll come to that in detail later, how it is being developed. Now, when you study about the cancer, you need to study about the right from the replication of the DNA itself. Actually, this portion is not there in the case written down, but if you don't understand this concept, it will be very difficult because there are two important functions. So you may be knowing the replication. There will be a leading strand. So when during the replication, the DNA will uncoil, and there will be a lagging strand and a leading strand. So this actually forms the leading strand and this actually forms the lagging strand. In the lagging strand, the synthesis happens in the discontinuous manner. So there will be the primers which will attach to the DNA. The fragments of primers will be attaching. Here there will be primer. The leading strand will go on continuously. That 5' to 3' synthesis will happen in a continuous way, whereas the 3' to 5' synthesis of the or during the replication is happening in a discontinuous way. Okay, so that is one point where the cancer is developing. Okay, now why the DNA replication is important is because of these two reasons. One is the proofreading function and the DNA. So if you, when you take the DNA synthesis, the DNA synthesis, as all of you know, it is done by the DNA dependent DNA polymerase, or generally we call it as DNA polymerase, which is responsible for the replication. Now, apart from the DNA synthesis, it is having two other functions. One is proofreading. Okay, it possesses 3' to 5' exonuclease activity. That means from the 3' to 5' in the leading strand, it can proofread the functions. Why the proofreading is important? Because the DNA dependent DNA polymerase is having, or it is an error prone synthesis. It is estimated that almost one error occurs when 1000 nucleotides are processed. So 1 in 1000, it makes a wrong base pair addition. So the part of the DNA replication, the replication enzymes, the DNA polymerase actually checks for the proofreading also. Okay, so it sees whether the exact matching is being done. If the exact matching is not done, what will happen? It will lead to a mutation. So it is a error checking mechanism to prevent the mutation. And as we see later, when a mutation is happening in the procarcinogenic region or an anti-cancer gene region, it can develop into the cancer, what you call as the initiation. So all of you will be knowing different states of cancer initiation, the clonal selection, propagation, and so we will see that later. Okay, so this polymerase is having another very important functions, which is the repair of DNA. Okay, again, this is a very important concept when we study about the chemical carcinogenesis, the DNA repair. Okay, so these are the two important functions of the proofreading and DNA repair. So the DNA synthesis. Then, as I already mentioned, we have the two genes. One is the procarcinogenic gene and next is the anti-cancer genes. We will see that later, see in detail about that later. Okay, now the mutations in the tumor suppressor genes or an overactive oncogenes may induce the individual vulnerability to the cancer. That is, when a tumor suppressor gene is suppressed and when a tumor promoter gene is promoted, then in both cases there will be the initiation of the cancer process. Okay, so remember that all the initiations do not end up in cancer because the body is having some other mechanisms to control or regulate the wrongly synthesized DNA materials. Okay, now these are the major procarcinogenic reasons, the genes, because this will not be there in the textbook. Okay, they will be randomly describing these genes, but I have made it into a systematic way. So these are the genes which are capable of procarcinogenic genes. They are otherwise known by the name oncogenes. Okay, so actually this procarcinogenic genes or the oncogenes are actually derived from the normal genes itself. Okay, they are known by the name proto-oncogenes. Okay, now again this term you will encounter when you read the cassette and all, but they may not be explaining it. Gene, one is the RAS family genes, and what happens is that is involved in the cell signaling. Now the mutated RAS, when there is a mutation that is occurring in the RAS, that can lead to a constant cell signaling for the growth and survival. So that is the one actually the leading to the classical outcome of the cancer, that is the uncontrolled multiplication. So that uncontrolled multiplication of the cell is actually produced by a defective RAS gene. Then MYC gene. MYC gene is actually responsible for the progression of the cell cycle. So when we study about the various phases of cell cycle, obviously all of you might have studied various. There will be a G1 phase. There will be an S phase where the DNA is being synthesized. G1 means this is the first growth phase. Then there will be an S phase, then G2 phase, then comes the mitotic phase. Okay, then another one which is very important in the breast cancer is the HER genes, human estrogen receptor 2 genes, which are amplified in certain breast cancers. Okay, it has a lot of implications because when we study about the various cancers like the triple negative, the positive and negative breast cancers, all this are involving in the mutation of the estrogen receptor genes. Now, another one is the gene, that is BCR-ABL, that is a fusion gene in the chronic myeloid leukemia, the CML. So usually it is the chromosomal translocation forms the fusion genes, which will activate the tyrosine kinase activity. When the tyrosine kinase activity is elevated, what will happen? There will be increase in the, or there will be increased phosphorylation of the enzymes. When there is increased phosphorylation of the cell, what will happen? The G1 and G2 phases of the cell cycle will be accelerated, and that will lead to an uncontrolled division of the white cells, which will lead to the development of the CML or chronic myeloid leukemia. Now, how the oncogenes are being activated? Here also the textbooks at various places they mention the oncogene activation, but really they don't mention what is actually the oncogenes. So how the mechanism is happening is by the point mutations that is occurring in the RAS system, or there will be gene amplification. So gene amplification means there will be multiple copies of gene will be produced. So the classical example is the MYC gene or HER2 gene. So generally there will be, say, maybe 4 or 5. So one gene may have multiple copies in our genome, especially in the eukaryotes. So that is one of the classical feature of eukaryotic genome, and that is the reason why the eukaryotic DNA is so bulky. Okay, as the evolution progresses, the number of copies also increases. Now, when multiple genes are created, what will happen? All the genes will start expressing. Okay, so instead of 5 genes, if an individual produces say 50 genes or 100 genes, what will happen? The stimulus will stimulate all the genes to express. So what will be happening? Multiple products or the same product will be produced in 100 times or 200 times in the increased level. Usually that is what is happening in case of the chronic myeloid leukemia as well as in case of the breast cancers. Okay, and as this process increases, the cancer becomes more and more fulminant cancer. Now, the next mechanism of oncogene activation is a chromosomal translocation. So that we have already seen how the from one sister chromatic, the chromosome may translocate to another or the other part of the chromosome. So that is known as the translocation from one chromosome or the one chromatid. It will be transferred to the next chromatid. So if this is the chromosome, which will be connected by the centromere, one portion may get translocated to this point. So that is known as the chromosomal translocation. So all these are important in case of chemical carcinogenesis. The next possibility is the virus-induced cancers. So usually it is a non-chemical method of transmission. So I'm not going too much into that one. But what happens is that all of you know that the virus will insert its own gene into the genome of the host. And if the insertion happens in the or in some activated region of the oncogene, the gene may become hyperactive. Okay. So that can lead to the development of a cancer. Okay. Now coming to the anti-cancer genes or the tumor suppressor genes. The tumor suppressor genes actually inhibit the cell division. They repair the DNA and promote apoptosis. So their function is to control the cell divisions when the cell divisions are exceeding a particular rate. Okay, they will be activated when the cell frequently divide. Okay. Now think of what will happen. Say suppose this is a particular gene and one portion undergoes a mutation. Okay, when that this portion has undergone the mutation, so what will happen? The protein synthesis will stop at this point. So you will get a truncated proteins. Okay, so this truncated proteins actually is dysfunctional protein. So what will happen? This unchecking mechanism or the checking mechanism for the uncontrolled multiplication will be stopping. So the most important one, as all of you might have heard of, is the TP53 or generally referred as the P53 gene, which is known as the guardian of the genome, which is capable of initiating the internal apoptotic pathway. And by the synthesis of the proteins, the pro-apoptotic proteins like the BAX, BAD, all this will be activated by the synthesis by the activation of this genome, and that will produce the apoptosis of the, or it will initiate the internal apoptotic pathway and death of the cell. So there are certain specific proteins which will act at particular cell cycles. One of the example is the RB1, which will regulate at the G1 phase of the multiplication. Now, these two genes are actually involved in the DNA repair mechanism. How the DNA is being repaired. Okay, so whenever there is an error, that error will be repaired by the DNA. The body has the ability to repair the DNA, which we will again see in the later on slides. Now, the APC is another cellular adhesion and signaling in colon, because this cellular adhesion is very important. As you know, the cancer cell can undergo, one of the most important or the striking feature of the cancer is that normally hepatocyte will not survive in the lung, but a cell that is having the hepatocellular carcinoma can get incorporated into the lung cells and can live happily there. That we call it as the metastasis. Okay, this is one of the striking feature of the metastasis is one of the striking feature of the cancer cell. Only the cancer cells are having the ability to metastasis. So that also is promoted. Otherwise, what will happen is that when a hepatocyte go to the lung, the hepatocyte and lung So you may be remembering the pathway, the integrin, the microtubule, and the cellular lamens pathway, which will actually communicate or help the cell to be at home. Okay, but when it is going to the lung, what will happen? It is a totally different environment. So the integrins will not be activated. So the cell will start collapsing. So that is what is happening in the cellular adhesion. But what happens in cancer cell is that this pathway is bypassed. This apoptotic pathway is bypassed in cancer cell, which is capable of metastasis. Okay, so that property is known as anoikis. I think all of you might have heard of that term. Anoikis means it is not in the home. Now, the DNA replication errors leading to the oncogenesis. Why I'm telling this is because in many portions in the textbook, they mentioned the terms without explaining what it is. Okay, first one is the base pair mismatch. Okay, so actually base pair mismatch, as the name indicate, there will be a base pair. So generally all of us know that cytosine will pair with the G and adenine will pair with the T. So that is what is normally happening in the cell. But what will happen when an adenine is paired with the guanine? So that is called a mismatch. So adenine is not generally, or adenine pairing with the uracil. So that is also, so generally uracil will not be present in the DNA. So this mismatches can happen in the while the DNA is being synthesized. So this is normally happening in the DNA. So don't think that only in the cancer cell this will happen. As I already mentioned, 1 out of the 1000 base pairs will be a mismatched base pair on an average. So there is a correction mechanism in the body. Now the aflatoxin, all of you know what is aflatoxin. This is a fungal toxin that is forming a DNA attack. So it can interfere with the base pairing during the replication. So remember, it is affecting during the replication. So that is the pathway. Actually, it is the not the B1 directly. It is the M1 and M2, which is actually causing the mutations, which is present in the milk and which is capable of inducing the mutations. Okay, so this mutations in the P53 gene can lead to the liver cancer. Okay, because P53 is a tumor suppressor gene. When the suppressor gene is mutated, the gene becomes inactive. It cannot further function normally. So that will lead to the liver cancer. Why it is specifically liver cancer in case of aflatoxin? Because that conversion of the aflatoxin to the epoxides are actually happening in the liver. So that is the reason why you get more cancer in the liver when the animal or the human being is affected with the aflatoxin. Now another mechanism is the replication fork stalling. Replication fork stalling means I have already explained the replication fork. So there will be a leading strand and there will be a lagging strand. Okay. Now what happens is that imagine that a nucleotide at this point is having something that is being attached, or the nucleotide has been separated like this. What will happen? The polymerase will come here up to here. The DNA polymerase will come up to here, but it will stop at this point. So this is actually what is meant by the term stalling. Okay, so that stalling, what will happen with the stalling? When the stalling happens, the full replication or the transcription will not happen. That is, synthesis of mRNA will not happen. The mRNA is synthesized by another polymerase, that is DNA dependent RNA polymerase, or generally we call it as the RNA polymerase. So the RNA polymerase cannot further act. It will stop at this point. So what will happen? You will get an incomplete mRNA. So this incomplete mRNA will be capable of will not be capable of synthesizing a protein that is to be required for the tumor suppression. Okay, so so if it is mRNA, the protein will not be synthesized. What will happen to the DNA in that case? So the DNA dependent DNA polymerase or the general DNA polymerase that will stop at this point. So what will happen? You will get a fragment of DNA. Instead of getting a complete DNA or complete genome, you will be getting a gap in the DNA. So this point will be omitted. So what will happen? The gene will be split in the middle. And when the gene is split in the middle and that is transferred to the daughter DNA or the daughter cell, what will happen? The daughter cell will not be able to synthesize that particular protein. So this is very important in the step of promotion of cancer. Okay, how the cancer is being promoted by the chemical carcinogens. This is very important. Now, next is the replication stress from the oncogene overactivation. When we discussed about the pro-oncogenes or oncogenes, we are seeing that these are the cells, these are the genes which will stress the cell or which will induce more multiplication in the cell. Okay, now how it is being done? It will produce proteins which are responsible for an increased number of multiplications. The cell or the molecules that will signal increased multiplication of the cell. So generally, when I explained about the MIC, I told you that it's the cell cycle progression. Okay, in the cell cycle progression, when these genes are overactivated, what will happen? The cell will go on multiplying uncontrollably. There will be more and more multiplication of the cells. Of course, it will be that will overload the replication machinery. And when the replication machinery is affected, what is the first thing that is affected? It is the proofreading done by the polymerization activity will be there, but the proofreading activity of the DNA dependent DNA polymerase will cease to exist. Okay, as I already told, 1 out of the 1000 base pairs is an error which has to be corrected, and it is a time taking process. So that will become unstable, and what will happen? The gene will be undergoing a mutation, and this mutation can lead to the oncogenesis. Now, first one is the next one is the unrepaired DNA damage. Already when there is a error in the DNA in the body, there are different mechanisms to correct a mutation. Okay, but when the cancer cells are being selected, so the next one, the next replication error that is happening in the DNA is the unrepaired DNA damage that I already explained. Next is the gene amplification error that we have already discussed. Certain portion of the DNA segment will be amplified to the development of oncogenes. Okay, so instead of 10 copies, there will be 100 copies or something like that. So what will happen? There will be that much number of products that is formed out of the amplification. So that is the next one. Next is the telomerase dysfunction. So all of you know what is telomerase. So when we take the chromosome or the DNA, at the end of the DNA, at both end of the DNA, you have certain repeated units, which is known as telomeric tandem repeats. Okay, this telomeric tandem repeats are actually essential for identifying how old is the DNA. If the DNA, so why it is like that is that this tandem repeats are lost during the proliferation or the replication of the DNA. So with each replication, some portions of the tandem repeats will be spliced off. So like ways. Now, why this is important? Because the efficiency of the replicating enzymes, the DNA replication enzymes, that is the DNA dependent DNA polymerase, when it is reaching to the end of the DNA, the replication efficiency or the transcription efficiency is very, very less. Or as it, so the same feature you will get with the when you do the PCR also. The amplification of the terminal sequences will not be proper. Okay, so some of the portions will be missed in even in the PCR. So in a very small, very specific genome, you are missing with the replication. Sorry, with the yes, with the replication. So imagine what will happen when there is a big chunk of DNA. So with every replication, there will be loss of certain portions of this telomeric tandem repeats. And when the total telomeric tandem repeats are reduced, actually this is a mechanism to protect the DNA from developing into a carcinogenic gene or a pro-carcinogenic DNA. Generally what happens is that when the DNA, the telomeric tandem repeats are reduced beyond a certain limit, the pro-apoptotic genes will be activated and the cell will undergo death. Okay, so this is a normal mechanism that is happening. Now, when the function of telomerase is actually to control the number of cell cycles that are being produced in the or the number at which the DNA can replicate. Okay, some DNA may be capable of replicating 10 times, 50 times, 20 times. That depends on the cells in which the DNA is residing. Not all the DNA is having the equal propensity for multiplication. So when there is reactive oxygen species, that will cause the damage to the DNA. So this is one of the mechanism actually leading to the cancer by the reactive oxygen species. Another mechanism of reactive oxygen species is the formation of adduct that again we will discuss. So when the telomeres are removed at a very fast rate, what it will happen is that it will form some ends which are known as the sticky ends. Okay, so one chromosome will be like this and another one DNA will be like this. So this is known as the overhangs. This overhangs will combine with this to form. So what will happen? This is the DNA fragment 2, 1 and this is the DNA fragment 2. Because of this overhangs, it is just what happens with the splicing, the restriction into nucleases, what we use for the introduction of new genes. It is the same principle, but happens due to a different mechanism of the reactive oxygen species. So this telomeric fusions will combine two DNA together, and when the two DNAs are combined together, that will become a dysfunctional because it becomes so big DNA. The replication cannot happen in a way with a high fidelity. Okay, so that is what is known as the genomic chaos, which is essential for the progression of the tumor. This is one of the reasons for the progression step of the tumor. Then we have already seen the DNA damage and mutagenesis, especially with the DNA adduct formations. Adduct formations, once again, we will see that can lead to mutations. So all this can lead to the production of the onco or the pro-carcinogenic cells. Okay, so the next one is the repair mechanism. There will be inhibition of the DNA repair pathways. So we will see that repair pathways in the coming slides. Next one is chromosomal aberrations, especially the translocations can lead to that previously discussed the telomeric fusions can lead to the genomic instability. Aneuploidy, when the number of the chromosome is variable, that also can lead to the genomic instability. So all this can cause the chances of production of cancer. Then cell cycle regulation. So cell cycle checkpoints, we have seen that various checkpoints are there. So when the G1 has to stop and the S has to start, when the S has to stop and the G2 has to start, when the M phase or the mitotic phase has to start, all these are having a cell cycle checkpoint proteins. So when the cell cycle check proteins are mutated, what will happen? There will be the cells will be present in all the phases. It can be in the G1 phase, it can be in the so in case of fulminant tumors, usually this checkpoints will be damaged. Okay, that is how the cell is capable or it is getting the property of uncontrolled multiplications that is happening in the malignant tumors. Then oxidative stress, again one of the major reason for the carcinogenesis, which can cause the oxidative damage to the DNA. So we will see what is oxidative damage when we come to the mechanisms. Next one is the epigenetic alterations. So all of you might have heard of the term epigenetics without understanding what exactly the epigenetics is. So when we say, or if you refer in text, it will say that there are different types of epigenetic variations. One is DNA methylation, then histone modification, then microRNAs. All these are coming in the epigenetic. So how exactly the epigenetic alterations are producing cancer, that we will see in the coming slides, because these mechanisms are not elucidated in the case written down. If you read, they will just mention that epigenetic alterations, but you may not understand how the mechanism, or when we go for the mechanism of individual carcinogenic agents, you may not be understanding unless you understand what is epigenetic alterations. Then apoptosis and cell death pathways, that I have already seen that one. Then the telomeric maintenance also we have already seen. Now, how the adduct formation, one of the major reason for carcinogenesis, the adduct formation is impairing the DNA pathways, or the DNA replication pathways, or the normal functioning of the DNA. Now, this is actually the structure of the DNA. So AIT pairs and GC pairs with a triple bond. So adduct means a molecule which is capable of binding with any part of the DNA. So it can bind with here, it can bind with here, and the adduct can bind not only with the one site, it can bind with the two or more site. So what happens is that when an adduct is formed here and when an adduct is formed here, they may interact, or one adduct itself will be capable of binding with this adenine and guanine. So what will happen when such, so this is known as the inter-strand adduct. Okay, now the next possibility is there will be an adduct which will bind with the A at this point and G at this point, which is known as the intra-strand adduct. So intra-strand means it is within the same strand itself. It will be, so if you take this, it will be in the 3' 5' direction. And if you take this particular example, the second one, the interstrand adduct, it is formed between two nucleotides in the two different strands. Okay, so here it is forming with the 3' 5', and here it is forming a bond with the 5' 3'. So it can cause the DNA distortion. Now, what will happen when the DNA is distorted, or how it is going to affect the replication? Now, the in the 5' 3' direction, the polymerase will be traveling at this point, or it will be moving at like this. But when it is reaching here, what will happen? This strand is already having an adduct or bond with this one. So when there is a bond, what will happen? There will be attractive force. So this, suppose this was the previous distance. Now what will happen? This A become very close to C. So what will happen? The double helical structure of the DNA will be lost, or the strands becomes more close together. So what will happen? This area will become lean. So what will happen? The DNA polymerase cannot attach at this point. So the transcription will stop at this point. Okay, because that angle of binding is also very important for the DNA dependent DNA polymerase. When that angle is lost, or the direction is lost, the DNA dependent DNA polymerase cannot act. So this is exactly how the adduct is causing a mutation. Now what will happen, or how it is producing the mutation? The DNA will stop at this point, which will induce a single stranded or a double stranded break in the DNA. Okay, when that break occurs, the protein synthesis, the uh, cell regulatory proteins will be stop being synthesized, which will lead to the carcinogenesis. So this is one mechanism. Next one is the epigenetics in the chemical carcinogenesis. I will explain it in detail. So the major mechanisms are the DNA methylation and histone modification. Non-coding RNA, especially the RNAs like the microRNA, siRNA, the small interfering RNA, and such mechanisms also exist. But for the chemical carcinogenesis, these two mechanisms are important. One is DNA methylation, and second one is the histone modification. Okay, now we will see what is meant by the DNA methylation and how it is a dangerous thing. So there will be the bases arranged like this. Suppose this is adenine, it will pair with the T, G, C. So the DNA methylation means a methyl group will be added to the 5' to the fifth carbon of the cytosine. Okay, so this usually happens maximally in the CpG islands, that is the cytosine, guanine cytosine rich or the GC rich areas. Okay, so CpG means cytosine will be paired with a phosphate group. The next nucleotide will be guanine. That is only the meaning, because in the textbook they don't describe what is CpG. So every nucleotide is connected using a phosphate bond. So that phosphate bond where the region will be rich with the C and G, that is known as the CpG islands. There are certain areas in the gene which are again regulatory, more of regulatory in nature, which is known as the CpG islands, and the methylation usually happens to the carbon. Okay, remember, it usually happens for the carbon that is involved in the cytosine. Okay, so this is known as the methylation, DNA methylation. Not all the methylation happens to all the nucleotides. Usually that happens in the fifth carbon of the cytosine nucleotides. Okay, and this is done by the DNA methyl transferase. And now, what is the reason for this methylation? As the DNA becomes older, more and more nucleotides will become, say, attached with the CH3 or methylated. So again, the more methylation indicate the older origin of DNA. Remember this point, because this point is very important when we come to the repair of the DNA. How the DNA repair mechanisms identify which is the old strand of DNA and which is the new strand of DNA. Yes. Okay. So this is how the repair mechanisms identify which is the old strand of the DNA and which is the new strand, because usually the error happens in the new strand. Okay, so when the repair has to take place, it has to identify which is the old strand. So we'll come to that when we come to the repair mechanisms. Okay, now when the CH3 group happens, it promotes the chromatin condensation. So this is one of the most important thing, making the DNA inaccessible for the next process, that is the transcription. The mRNA, the DNA dependent RNA polymerase cannot attach to the DNA once there is condensed. So when you studied, you have studied the euchromatin and heterochromatin regions. So more area becomes the euchromatic area. So the RNA dependent, so DNA dependent RNA polymerase cannot synthesize the sufficient quantity of mRNA. Okay, now it can lead to a hypomethylation state as well as a hypermethylation. Means there will be hypomethylation, means rather than having a particular number of methyl groups to be added normally, the number of CH3 groups that is attached will be very less. Now again, that is also important, because What will happen when the CS3 is reducing? More number of genes are going to express. So instead of, say, I already told you there will be multiple copies of DNA, and this multiple copies of DNA some of the DNA will be suppressed from transcribing with the help of methylation. And when that methylation is removed, what will happen? All the genes will start expressing, which will induce a increase in the number of cell multiplications in the tumor progression. Okay. Whereas the hypermethylation, in hypermethylation, what will happen? More DNA will be condensed to the chromatin. So that will lead to the silencing of the gene. As I already told, when there is condensation, the DNA becomes inaccessible for transcription and translation. Okay. So when a hypermethylation is occurring in the tumor suppressor genes, those genes will not be expressed. So what will happen? The there will be a clonal selection of the cells which are hypermethylated. Okay, so this is one of the mechanisms induced by the polycyclic aromatic hydrocarbons, arsenic, vinyl chloride. All those compounds are capable of producing DNA methylation. So this is what is happening, the methylation. So this is how the methylated cytosine be there. So usually this methylation happens in the promoter region. So when the promoter region is affected, so this is the unmethylated, there will be normal transcription and the translation process. Translation process. But when the promoter is methylated, what will happen? There will not be any transcription. So the cellular checkpoints will be affected. The cells will start multiplying at a faster rate. So when it is happening in the tumor suppressor genes, these proteins are essential for suppressing the cell growth. In the absence of that suppressing factors, the cell will start multiplying uncontrollably. So this is how the DNA methylation is producing carcinogenesis. Next one is the histone modification. Now, histones are the proteins that are seen around the DNA. Okay, so like this, the DNA will be wound around the histones, the DNA fragments, or the DNA strand will be wound around this histones to conserve the space, as well as, now what happens is that the DNA that is bound with the histone, they do not undergo a transcription. Okay, it will not be converted to the mRNA. So the more DNA is attached to the histone, the less number of transcriptions will happen. So this is a part of gene silencing and gene overexpression. So when the histone modification or when the histone acylation happens in an excessive rate, the genes will be silenced. Some genes will be silenced. When the histone deacylation happens, the more number of or more part of the DNA will be not winding to the histone core. What will happen then? There will be overexpression of the certain genes. So this is how the histone modifications are happening. Now these are the examples of common histone modifications. Then types of the DNA damage. The DNA alterations caused by reactive oxygen intermediates or the chemicals. So the common damage type will include at 3 points. One is the base, the nucleotide bases, then sugar and the phosphate, because these are the 3 parts of the DNA. Attack can bind with any portion. It is not that it will bind only with the nucleotide. It can bind with the phosphate bond. It can bind with the sugar, deoxyribose sugar also. Okay, and the DNA cross-linking can occur with a bifunctional alkyl. Bifunctional means it will have 2 polarity. One will bind with the one strand, another will bind with the another strand. Or sometimes what happens is that it may bind with the same strand itself, which is known as the intrastrand adducts, and this is the interstrand adducts. Now what happens is that the polymerase, when it is coming for replication, when there is intrastrand, what will happen? There will be a break in the one strand, and if it is an interstrand, what will happen? That will lead to the development of a double-stranded break in the DNA. So this is how the single strand and double strand breaks are created within the DNA. Okay, so this is again happening when there is the mustard, the alkylating mustards, which are used as anti-cancer drugs. And again, UV irradiations, ionizing radiations, all this are capable of producing the single-stranded or the double-stranded modification. As the energy increases, there will be more number of fragments. Say when it is exposed to gamma ray, the number of breaks will be innumerable. So the total DNA will undergo the lysis. Again, that is the mechanism by which we use the radioactive material for the treatment of cancer. The entire genome will be blasted into pieces. Okay. The DNA polymerase proofreading actually corrects the many replication. And this chemical induced changes, certain chemical induced changes are repaired in an inefficient way. Okay, and if the damage is not repaired before replication, what will happen? That will develop into a mutated cell or the cancer cell, or the that process we called as the initiation in the cancer. Okay, now these are the various different various types of the cancers or the damage which has been given in one slide. One is it can form adduct. I have already mentioned the adduct about the then interstrand and intrastrand crosslinks. Then mismatch is another problem. Generally, the G has to pair with the C. So instead of pairing with the C, it has been paired with the uracil. Uracil is an uncommon nucleotide in DNA. Usually, it is seen in the RNA, but such mismatch also can happen. Now, this is an example of a single strand break, and this is a double strand break. Now, there is a more important damage which is known as the helix distortion. Now, what happens is that when the adducts are formed, as I already told, the angle of curvature of the DNA will be changing because of the attractive forces, which will lead to the helix distortion in the enzymes. So generally, what happens in DNA is that this portion of the DNA will be spliced off and it will be resynthesized. Okay, so that is what is happening. We will see how that is being achieved also. This is done by the mechanisms of DNA repair mechanisms. Now, there are certain pathways. Again, these pathways are not mentioned in the textbook, but frequently they refer about the base excision, nucleotide excision, thinking that the readers know it. So that is why I'm explaining this. Unless you understand these terms and the differences in the terms, it will be very difficult to comprehend what is written in the cassette and out. Actually, this is actually in the textbook. It is as per the textbook, but how the mechanism is there, they are not mentioning it. Okay, first one is the direct DNA repair. That is a one-step DNA repair. It actually corrects the damage without removing the DNA strand or replacing any nucleotide. Okay, it is a spontaneous recovery method. Okay, which actually involves two pathways. One is the methylguanidine DNA transferase pathway. Okay, next one is alkyl B. So I'm just mentioning that pathway. There is no need to specifically learn these things. Only just understand that there are two pathways. Okay. Next one is the base excision repair. Now, in the base excision repair, what happens is that so this is what is happening in the base recognition. First, the damage is being recognized. Okay, so here you can see a uracil. The uracil is not actually incorporated from the cytoplasm, but it is occurring by the deamination of the already existing cytosine molecule. Okay, but the presence of uracil in the DNA is a mutation, or it's an abnormality. So actually the uracil DNA glycolase enzyme identifies and remove the abnormal base pair. What will happen then? There will be an epiurinic or apyrimidinic site or AP site. That means in this portion there will not be any nucleotide. So G will be unpaired here. So that is known as the AP or the epirinic or apyrimidinic site. It can be a purine base or a pyrimidine base. So the corresponding nucleolase will cut, or the endonuclease will cut that mismatched pair, and the phosphodiesterase will cut the sugar backbone. The sugar backbone here is also cut. So what will happen? It will end up in a single stranded nick. Okay, and this nick will be filled by the DNA polymerase beta. This gap, because there is only one nucleotide that is missing. So that will be easily repaired by the DNA polymerase, which will incorporate instead of uracil. The uracil will be excised by this enzyme, which will produce a cleaved site, and the cleaved site will be processed by the gap filling enzyme, the DNA polymerase and the DNA ligase. So this gap will be remaining intact, and this will be resynthesized, or the resynthesis or the end will be sealed by end of the sugar molecule will be done by the DNA ligase enzyme, because the polymerase can provide only the nucleotide linkages. The backbone should be corrected by the ligase enzyme. Okay, so when that is occurring, it will finally end up in the, so the damage will be replaced, or the damaged area will be. So why it is called as base excision repair is, it is a single base that is excised off and repaired. Whereas when it is coming to the second repair mechanism, which is known as the nucleotide excision repair, or NER, it actually removes a bulky or helix distorting. So we have already seen what is helix distortion, and there are two types of NER. One is the global genomic NER. That means it will scan the entire genome for the damage, where there is any damage in the helical or whether there is any distortion in the helix. Next one is transcription coupled NER. So this usually is activated when the transcription is occurring. Okay, see the mechanisms of NER. So this is actually the mechanism. It's a little bit complicated mechanism. So I'll represent it into. So this is the transcription based correction, and this is the global genome repair mechanism. So whenever there is a DNA damage which involves multiple nucleotides, it will be recognized first by various proteins. So after the recognition, what will happen is that this recognized site will unwind. So this is, imagine that this is the damaged site which causes a distortion. So what will happen is that because of these enzymes, it will be unwound, and after unwinding, various proteins will act on this portion and will cut the DNA. So cut and remove the DNA. So among this, one will be the some area will be the place where it has formed an intrastrand or interstrand adduct. Okay, so when it is cutting for a long distance, whatever the damages that has been happened, which causes the strain in the DNA molecule, will be removed, and the same nucleotide will be resynthesized. And again, once the gap is filled by the DNA polymerase, there will be ligation. Next one is the mismatch repair. Now, in mismatch repair also, it is again mismatch is being detected here. So there will be a distortion in the DNA molecule, but the difference is that in the strand discrimination. Okay, here both the strands will not be recognized. I told you previously how, say, imagine that this is the parent strand and this is the daughter strand. How the repair mechanisms identify it as a parent strand and daughter strand? It is by counting the methylations. Okay, the one with the more number of methylations will be counted as the accounted as parent strand, and one with the less methylation will be counted, because the methylation increases when the DNA becomes older and older. Okay, now this will be used as the, so in this mismatch repair, this will be taken as the parent template. Okay, and this area only will be excised off and the complementary strands will be added. Okay, so here the resynthesis happens according to the parent strand. The daughter strand is synthesized and it will be ligated. The next one is homologous recombination. So in homologous recombination usually happens in a double-stranded nick. Okay, now a double-stranded break is recognized by certain proteins. Okay, and what happens is that this repair mechanism will cut a portion from here and cut a portion from here. So what will happen? There will be formation of a structure which is known as the there will be yeah. So actually it has to pair from here, but up to this point it will be unpaired. Here also this portion will be unpaired. Up to here it will be paired, and this is known as the overhangs. So this overhangs, all of you will be knowing when we study the genetic engineering, we create sticky end with the restriction endonucleases. So likewise, there will be sticky ends. So what will happen? It is not directly being attached with each other, but what happens is that now here there will be two chromosomes. It may be attached at the time of mitosis. It may be attached by the centromere. Now this portion is being nicked off, but that this is the portion that is. So it will search in the sister chromatid for actually what is the base. So the error checking is not done within the strand. It is moving outside the strand, which is known as the strand invasion. Okay, the strand invasion is actually, or it will produce the sequence. So if this is mutated, the complementary sequence will be taken from here. Okay, and the DNA structure will be finally restored. So this is the invaded strand, and based on the sequence in the same gene in the sister chromatid, so that is why it is known as the chromatid or the sister chromatid exchange. So here this portion is actually based on using the template at its sister chromatid. So there is a problem in that also, because if this is mutated, this one also will become mutated. So if a heterozygous individual, there will be mutation here, this area will be unmutated. But unfortunately what happens is that when this is being used as template, this also will become mutated, which will precipitate the cancer in case of homologous recombination repair. Then next one is non-homologous end joining. So in non-homologous end joining, again also it is not depending on the next daughter strand. So what happens is that this is usually happens in case of the higher mammals. So this will be the nicked end, and this nicked end will be end processed using certain proteins. So it will become a uniform one, and this end process will be sealed by certain enzymes, which are the Artemis and other enzymes are there. No need to bother about this enzymes actually, because that is not given in the textbook. And the gap will be filled, temporarily filled by a logic. Okay, it is not depending on the strand in the sister chromatid. It is based on a logic. So the non-homologous end joining can produce errors when compared to the homologous end joinings. So the end will be ligated and the new strand will be synthesized. Okay, so this is possible when both the strands are not damaged. Okay, so this is one disadvantage with this non-homologous end joining. I hope these mechanisms are clear, because for moving further in the next classes, you will require, or if you are reading the textbook, you will need to understand this concept. So if that is again, if anybody requests the slides, I can provide it to the organizers so that they can distribute it, because many of the slides are not there in the textbook, or the mechanisms are not mentioned in the textbook. But to understand the carcinogenesis, you need to understand these mechanisms.